Hey guys, I'm back with another cell activator experiment. In this series, I'm testing out several different additives in Floetrol-based acrylic pours to see what type of cells we can create. I wanted to see what else, other than silicone, could be used to produce cell action in Floetrol pours. Today I'm testing out good old WD40. I'm using a mix of around 90% Floetrol to 10% GAC 800 in these experiments. You don't necessarily need the GAC, I just add it for the extra protection from cracking as the paint dries. If you've missed the previous experiments, I'll leave a link to the playlist for you in the description below. I added 2 drops of the WD40 to all of the colors except the base white. From there I layered each color into a separate cup and then flipped that over onto the canvas. This method is called the flip cup. It's pretty great for cell production as it causes quite a shift in the paints. The paints moving over and under each other is the main cause of cells in acrylic pours. I'm describing the cells from the WD-40 as broccoli cells. The shape that some of them took on reminds me of broccoli florets. Like in the pour with the mineral oil, there's this oddball circular shape that appears stain-like. There are cells within this weird area, but something about the shape of it is so inconsistent with the rest of the paint that it seems odd. I love the way the cells burst open to show layers of color though. If you've been keeping up with this series, you'll notice that the majority of these results have a high concentration of red paint. It comes down to the paint transparencies. The Cadmium Yellow Light is a transparent color, the Ultramarine is a semi-opaque color, and the Pyrrole Red is an opaque color. If I would have picked a red that was a transparent or semi-opaque, they wouldn't have come out quite so red dominant. Alright guys, I'm going to leave you to the rest of the experiment. Don't forget to stick around to the end to check out the close-ups of the dry results. In the next video, we're combining the last 4 test subjects into one pour. After that, we're moving on to the next 4 additive tests, so make sure you go ahead and subscribe now so you don't miss the next one. If you have any questions or recommendations of additives you would like to see tested out, you can leave me a comment below. I'll see you guys next time. Bye!